Well, unlike the previous talk, I'm entirely science. It's a very discovery-based presentation. And unlike the previous talk, the slides aren't really set up for the speed dating format. So I'm going to have to zip through and hit some of the high points. So the platform we're developing at Rise is based on trying to make huge libraries that share a lot of features with cyclosporin. Cyclosporin's the only orally available peptide of any real size on the market. And what makes it special, among other things, are the fact that it's cyclic, it's relatively small, it's 11 amino acids, it has multiple N-methyl backbone substitutions, and it has several non-natural amino acids. And so what we want to do is make enormous libraries of things that look like cyclosporin. The libraries tend to be less lipophilic than this and have a little more chemical functionality. So lots of isosteres and heterocycles, things like that are common side chains we like to use. And the way that we want to set this up is so that we make these enormous libraries and then screen them directly for function in this format. So the technology we use is called mRNA display. And mRNA display is similar to phage display in that you get to pan against a target of interest, wash away the things you don't want, and amplify the high affinity binders to that target. And the way you set it up is that you start with a DNA pool, which has an open reading frame that would code, in this example, for a peptide of this format. You transcribe that to mRNA, and you link puromycin onto the end of the mRNA. Puromycin, through some chemical trickery, is able to chain terminate a ribosomal peptide synthesis. That's how it works as an antibiotic. But when you put it on the end of an mRNA, it tricks the ribosome into passing the peptide it was synthesizing and attaching it onto the mRNA it was reading. So now you have a phenotypic, the peptide, to genotypic linkage, the mRNA. We then cyclize the peptide moiety of this conjugate chemically through an alkylating agent. We use, in the examples I'm showing today, dibromoxylene, which is very efficient and works well in our library formats. And then we reverse transcribe the mRNA. So this is a cyclic peptide displayed on an mRNA that codes it, and then this complementary cDNA is also present. And this undergoes selection just like you would do in phage display. Following that round of selection, you use PCR to amplify the winners, and then you can continue this for multiple rounds to enrich the pool that started on the order of 10 to the 12 sequences, enrich that in binders, and then you read out those sequences by DNA sequencing. So the way that we get the unnatural stuff in here is that we reconstitute this in vitro translation reaction. The peptide synthesis reaction, is done in a mixture where we control all the amino acids that go in, and we can leave things out as well. So a lot of natural amino acids are undesirable for cell permeability. We omit those and replace them with things that are more compatible with cell permeability. And then just to make it abundantly clear, we're reading DNA sequences, then we go to a peptide synthesizer and synthesize that molecule. I'm going to skip this one and get to this one. So this is a slide that summarizes one of our more advanced programs. This is an undisclosed circulating protein-protein interaction. In this case, we're looking to come up with a synthetic replacement for a monoclonal antibody. So directly from selection, this is the top left here, these are our hits that we get out of this screen. So these are 13 amino acids. We've read DNA, gone and made them. And with an FP assay, you get a biochemical KD or Ki of 7 to 10 nanomolar. We look in a functional assay at the biological response. We see a similar IC50. And then if you look at plasma stability, this is really where this unnaturalness becomes important. So the peptides, the direct hits that we're selecting have a plasma stability on the order of 20, 22 hours. For demonstration's sake, we did the same selection, this mRNA display, with natural amino acids, just to show you what the special mixture of amino acids we have is giving you. And those are gone in like 8 minutes. So the the ingredients in this library really are giving you directly through selection in a process that takes a couple weeks, potency and stability in plasma. And then to go from here, this plasma stability to in vivo PK is on the order, in this particular example, of about 50 compounds that we made. And you can see through a couple, there's 2 peptides here. They're actually not that different from one another. And we're seeing IV half-lives of 5 and almost 4 hours, and a sub-Q of 5 hours. And in this particular project, the target concentration is around 1,000 nanograms per ml. So, and most of these, especially this dosing regimen, we're getting target coverage out to 24 hours. So one of the, I know this year's theme is peptides in oncology. So we have an MCL1 program. MCL1 is a member of the BCL2 family. These are anti-apoptotic proteins, and BCL2 and BCLXL have been drugged very well through fragment-based methods, but MCL1 has sort of thus far been resistant, and it's an important member of this family. It's a driver in several cancers, and it's also a resistant mechanism to several therapies, including BCL2 inhibitors. So we wanted to develop a selective MCL1 inhibitor. This is our hit compound in this project. By SPR, we are getting a KD for various compounds of 11 to 150 nanomolar. When we look in a competition assay with the helix from the protein from the interaction partner that this protein binds to, so this is really a functional assay in this case, we get IC50s on the order of 300 nanomolar. We have a crystal structure of this bound to MCL1. So the green is the physiological substrate or interaction partner with MCL1. It's a BH3 helix. And you can see that from this library, we found a motif that you would never design to bind this site. This site's evolved to bind helices. And we get this really unusual beta turn. And then through a few iterations of sort of rational design from here, where we can get the KD and potency improved to the point that we can start to see cell activity. And the readout we're using here is caspase activation. This is our first attempt at a cell assay. It's not really the greatest for this because this cell line is actually, the caspase is dependent on BCL2, XL, and MCL1. So we need to get these compounds into a cell line that are more dependent on MCL1 only. We also have a RAS program, and I'm going to assume that the audience knows the importance of RAS. RAS is one of the undruggable targets that everybody would like to gain some traction on. So we did a selection on RAS from that same library we used for MCL1. This is a cartoon to kind of give you a flavor for what's in there as best I could. It has a couple natural amino acids. It has 3 N-methyl positions and several unnatural side chains, being the red, and then one of these is an N-methyl and unnatural. So we did this selection against HRAS. HRAS and KRAS are biochemically very similar. We got very tight binders to HRAS, and then we were able to show by SPR that you're looking at a KD of about 3 nanomolar to KRAS. And remember, these are selections done based on binding, so we don't know if these are inhibitors yet. When we look at functional assays, the normal functional assays you would use for determining if you've inhibited RAS, we don't interfere with the interaction with RAF, and we looked at that in 2 ways, and we don't interfere with the GTP exchange of the protein. We have a crystal structure of this compound as well, and it actually makes a very similar secondary structure, and interestingly, it binds a surface that nobody really knows what it does. So the location of this site is consistent with the biochemical data, and we feel that you really need to be looking in cells to find out if this peptide is able to do anything. we've been able to show that this compound is cell permeable, and I'll sort of take my word for that. And then we've looked in at least one cell line, H358s. This is a KRAS-driven cell line. And at high concentrations, we start to see cell dying out at 72 hours. You can see that graphed here. What we're doing now in RAS is we're trying to take what we think is an unprecedented tool, something that gets in cells and binds RAS very tightly, and trying to either use it as is or convert it to something that inhibits RAS, either by mislocalizing RAS or sort of modulating it in other ways. We have a great set of academic collaborators, RAS experts, that really believe that we have something special and are going to help us turn it into a true inhibitor. The last thing I'll leave you with is, I started talking about cyclosporin as being something special that we want to make libraries that look like. This is an overlay of cyclosporin. This is the membrane permeable conformation of cyclosporin in cyan with our RAS and our MCL compounds. And the backbone of these are very, very similar. The side chains are totally different, but we're conformationally getting things out of these libraries that do the same thing as cyclosporin, and that may explain the cell permeability that we see. So leading into the discussion, I'll just put this slide up. All the information on this slide is available on our website. And with that, I'll take any questions. Yes.
It would be great to use a screening method to find peptides that have good cell penetration or oral bioavailability